In this tutorial, you will learn how to run the polymerase chain reaction, or PCR machine, to amplify a specific segment of DNA for further analysis. Human, alligator, and cockroach DNA samples have already been prepared with the correct primers and reagents, and the samples are now in the tube rack. Open the PCR machine by clicking on the top of the machine. Drag the Eppendorf tubes from the rack into the machine and click the lid to close the machine. Click the green arrow to run the machine. You can click the PCR control projector to view details of the polymerase chain reaction as well as viewing a representation of the reaction itself. The PCR will run through a three-step cycle of denaturation, primer annealing, and elongation, where each step occurs at a specific temperature for a specific time. In this tutorial, it will run for 35 cycles. Click the lab clock to advance the time by 3 hours and 10 minutes. Notice the status of the reaction in the LCD panel. After advancing 3 hours 10 minutes, the LCD will show that the reaction is complete after running through 35 cycles. After the PCR is complete, open the lid and move the tubes from the machine back to the tube rack. The amplified samples are now ready for further analysis. This concludes this tutorial on running the PCR to amplify DNA for further analysis.